Welcome back, everybody, from lunch break. It's a great pleasure for me to introduce my co-chair, Merab Kokaya, who is a professor of neurophysiology, also the chair of the Epilepsy Center at Lund University. He's also known as the coordinator of EpiTarget. Go ahead.
So, welcome everybody after the lunch with a new energy. Felix and I tried to put together a very short introduction based on the theme of the session, but also the presentations that we are going to have. So, therapeutics, what are the aims and what are the gaps? And we listed a few of them here. The aims are that we want to have seizure-free patients living a full life with a good quality of life and social participation. And the gaps are that we need to understand better the mechanisms of ictogenesis and epileptogenesis to address that. Increase efficacy and tolerability of the therapy, so better therapies. We need to have novel disease-modifying treatment strategies. Reduce morbidity and mortality in epilepsy patients. And of course, we also need to develop novel personalized treatment strategies. And it has been a lot of discussions about how to reduce stigma, and we have to do something also in that area, but perhaps in collaboration with the other researchers that have this topic as a specific research topic. So we know that there are challenges in the treatment of epilepsies. There are new drugs coming all the time, like shown here. But the efficacy still remains unimproved, and it's mostly seizure suppressive. Here is the example of one of this type of comparison between sort of standard drug and the new drugs, and we see that they are not statistically significant from the basic drug. So we need more breakthroughs, I would say. Cure and disease modification is incomplete at the best, and perhaps more personalized approaches will help us to develop this type of strategies. For example, resective surgery, in this case, gives perhaps 70% of seizure freedom. How long-term this is, it still remains to be, it's still debated. The study shows that different doses of mTOR pathway inhibitor sort of decreases the seizure frequency over time. But again, if you remove the drug, we don't know what happens. But there is some indications that we could be in the right direction. So current status, and also based on what we are going to hear today. So we will hear about the epileptic encephalitis. We will hear about the preventive treatment strategies that are emerging currently. We will hear about some novel screening designs in preclinical studies and multi-targeting drug combinations, as I mentioned previously in my introduction. We want to understand better the epigenetic control, and we will hear about that as well. And also other novel targets that are emerging, for example, neuropeptides, neurotrophic factors, and so on. Also, the therapies in the future, perhaps modification of the tissue, like gene and cell therapies, chemo and optogenetics, and perhaps less invasive tissue resection deactivation with the novel approaches here. So, how to fill the translational gaps? We need close cooperation between experimental clinical researchers, which is a standing sort of point to be addressed. We need better phenotyping and biomarkers. We talked a lot about this already, and we will continue to talk, and perhaps stratification of the patients on the risk after the brain insults. Data sharing, databases, multi-center, multidisciplinary studies, and perhaps this meeting will facilitate this type of approaches among ourselves. We need also kind of long-term funding for translational back and forth studies, which we are lacking perhaps today. And somehow we need to put epilepsy on the agenda, which we are trying now to do. Perhaps more importantly, get the big industry involved, including EMI. We have mentioned that already today. And if not, maybe we should be more daring to go for startups and spin-off companies. And perhaps also we need to develop more models that would address the pharmacoresistance. So I think this was our last slide. Okay, it's not moving, so I assume it is. With this short introduction, I would like to announce the first talk in this session given by Fabio Benvenati from Istituto Italiano di Tecnologia. Is it correct? Fabio.
So thank you very much for the invitation. I will talk about innovative therapeutic strategies for untractable epileptic encephalopathies. That is part of the Desire project, and one work package is devoted to this, and I will show you some of these results that we have obtained. Of course, these are ideas in progress, and they are pretty far from clinical application, but still could be an interesting field to exploit. What I will talk about is mostly an optogenetic approach, both for silence neurons in order to shut off a seizure, a similar optogenetic approach to regulate gene transcription. Particularly, we focused on the transcription repressor factor REST that is increased during epileptogenesis, and a third, a cell therapy approach to direct reprogram GABAergic neurons and to implant these neurons in the epileptogenic tissue. Okay. Let's start from the first one. So the idea was since during seizures and hyperexcitation state, there is an acidification both of the cytoplasm of neurons and the extracellular space, we thought to make a sort of a nanomachine that endows neurons the possibility to sense the drop in pH and to decrease the activity. So first we engineered a pH sensor that is a mutant of GFP, which has a very nice working range in its fluorescence emission when it is activated at 408 nanometers. And you see that there is a high dynamic range between the neutral pH and acidic pH that can be reached in an hyperexcitation state. Here, for example, are neuronal networks, primary neurons, subjected to bicucolin, so simulating in vitro seizure, and the black trace is the pH shift that goes up to 5.8. Okay, so the idea was to make a chimera by putting together the pH sensor and an optogenetic probe, a well-known one, halorodopsin, that is has an activity to silence neuron because it's indeed a chloride pump generating an outward current that hyperpolarizes the neuron and silencing its activity. However, of course, GFP-derived sensor has to be excited by exogenous light, and this is pretty invasive and also localized. For this reason, we thought to to make the probe even a little bit more complex by attaching an endogenous source of light that is a mutant of luciferase that can be activated by assumption of the substrate that can be assumed peripherally or per os. So in other words, you have an internal source of light that you can activate with some pill. And importantly, this construct, in this construct, the luciferase will activate GFP, and the fluorescence that is highly increased in acidic pH will activate the inhibitory sensor, the inhibitory opsin. This is the construct, so the luciferase for bioluminescence energy transfer excites GFP, the sensor, and GFP emits as a function of pH light, which activates in turn halorodopsin and brings about the inhibition of neurons. This sensor, the complete sensor, is expressed very well on the membrane thanks to halorodopsin. Without halorodopsin, it goes into the cytoplasm and induces, by administrating the substrate for luciferase, a very nice time-dependent hyperpolarization, which is completely absent at neutral pH or slightly alkaline pH. And as you can see, the acidification can reach over 15 millivolts, which is a noticeable signal to silence the neurons. Interestingly, we obtained acidification only with the substrate that induces violet emission of luciferase and not with an alternative substrate by which luciferase emits in the blue range. So it's also specific for the substrate in order to have breadth and fret that makes the probe working. Okay, the second topic, I'm sorry to go very fast, but just to give an overall picture, the second topic regards the dysregulation, the transcriptional dysregulation that, as we have seen very often, is associated with epileptogenesis and also thereafter to the overt phenotype. In our case, we focused on a transcriptional repressor, REST, that is very interesting because it controls among about 2,000 genes, but all the relevant neuron-specific genes. So in other words, it controls the neuronal phenotype, and the levels of REST and the activity of REST are inversely proportional to the expression of the neuronal phenotype. So the strategy was that, so since REST controls neuronal phenotype, and the REST level very often are dysregulated or mostly increased in several brain diseases, and including Huntington disease was one of the first diseases known for REST dysregulation, but also epilepsy, stroke, and multiple sclerosis. We decided to make an optogenetic probe acting intracellularly, which modulates on demand the activity of REST without altering its transcription and translation. So this is just to show you that the model that we chose and from which we were inspired is the cainic acid-induced temporal lobe epilepsy, in which you have an increase in REST. This is the control, and this is after administration of cainic acid in the hippocampus. So we have an upregulation of REST in this model. What we in order to have an optogenetic probe working on demand, we basically used a protein module. Is called love domain that comes from a plant, Avena sativa. And this love domain is interesting because it has an alpha helix in the dark, which when it is illuminated at appropriate wavelength, it becomes unfolded. So if you attach to the terminus of the alpha helix whatever inhibitory peptide, you have a sort of a molecular switch in which the peptide is hidden in the dark and becomes exposed and active in the light. And the idea, we tested several peptides, but particularly we are using this peptide that inhibits the interaction, the association between the cofactor MSyn3 and REST. And without MSyn3, REST loses its transcriptional repression. Okay, very fast. Well, these are the, by activating this probe, we see an upregulation of the REST target genes that we found also in neurons, for example, sodium channels or BDNF. And when we are in the preliminary stage in the molecular model of kainic acid-induced seizures, in this case we inject the probe before the kainic administration to see whether an inhibition of REST ameliorates the phenotype. Here we see that the probe injected in the hippocampus doesn't change the expression of REST, but increases the expression of REST target genes. And what we found is that the overall seizure score, as well as the more severe seizure types induced by peripheral administered kainic acid, are significantly ameliorated in these animals expressing this probe, as well as also the latency for the more serious seizures. The very last approach I want to talk about is reprogramming. So as you know, in epileptogenesis and epilepsy is sustained very often by an excitation inhibition imbalance, and very often due to a decrease in inhibitory strength. So one way to increase the inhibition and controlling epileptogenic networks is to transplant in GABAergic interneurons. We can easily obtain GABAergic interneurons by reprogramming iPS cells obtained from fibroblasts of the patients, but this has some risk for transplantation because of the possibility of proliferation. Then a much safer system is to take the same fibroblast, but without going through the iPS stage, the stemness stage, reprogramming them directly into a specialized element that is a neuron. So this study was done by Vanya Broccoli in collaboration with us for the physiological part. In this case, you see that the GABAergic neurons are reprogrammed because the GAD1 the GFP under the control of the GAD promoter is lighting up. This is a genetic screen showing that from the starting material, skin fibroblast, you get IGABA neurons, so reprogrammed neurons that have a transcriptional profile very similar to bona fide embryonic neurons. And they, in vitro, they undergo a maturation with an increase in the firing rate, and they can reach also very high firing frequencies. For example, here is over 70 Hertz, that are very typical of parvalbuminergic interneurons. And when they are plated on a feeder layer of normal neurons, they undergo synaptic connection with them. In this case, you see there is the reprogrammed neuron is postsynaptic to a normal neuron. In this case, instead, makes the presynaptic counterpart. The transplantation of these neurons was successful in the sense that the neurons survived to a large extent and could be around for several weeks, up to 8 weeks, we checked. And although most of the neurons stayed very limited to the area of injection. Nevertheless, they formed synapses with the host tissue, and since we labeled them with channelrhodopsin, we could stimulate them selectively. And so what we found is that by patching the reprogrammed neuron, we found excitatory and inhibitory spontaneous currents from the surrounding tissue, surrounding neurons, as well as by patching a host neuron, we found inhibitory currents induced by illumination that were abolished by bicuculin that basically prove that a synapse was done by the grafted neuron on the host neuron. Okay, what are the challenges and the next step? Regarding transplantation, the problem is to succeed in getting long-time survival and to have a spatial migration that is not limited in a few hundred microns with respect to the injection site. The other possibility is instead trying to make gene editing of the endogenous neurons in order to increase the excitatory strength, an alternative method. Regarding the optogenetic regulation, this is the optogenetic probes. The bottlenecks are those of any gene therapy approaches. I've shown you that going from optogenetics to chemoptogenetics for sure decreases invasivity, so there is no need to illuminate. The problem remain regarding the adenoviral vectors, anyway, the viral vectors to express and these probes in neurons and the potential immune reaction. Now we have very interesting adeno-associated virus that can infect neurons after intravenous injection, and of course an effective targeting to the epileptogenic population. Okay, so with that, I thank you for your attention.
To now introduce Emilio Peroca. Where are you, Emilio? Is he here? Ah, there. Very good. He's our past president of the ILAE. He's also a professor of clinical pharmacology at the University of Pavia, and he's running the Mondino Neurological Institute Clinical Trial Center, if I'm correct. Thanks for coming.
Perfect. Thank you, Felix. And good afternoon to everyone. Thank you for the invitation. It's wonderful to be able to really get a glimpse of what's going on in terms of research in Europe these days. The title of my presentation is a little bit different from the previous one, and it has to do, of course, with translation. How all the advances from this research, which ultimately have to benefit people with epilepsy, are going to be brought to fruition. It has to be done, of course, through clinical trials. Am I back to? Oh, maybe this should be the one. Okay. So just to remind ourselves, clinical trials are crucial on 2 points. First of all, we need clinical trials to demonstrate efficacy and safety for any compound that develops out of the efforts from basic science and bring that to the benefit of the patients. Secondly, we need clinical trials to inform rational prescribing clinical practice. And one issue that we have at the moment is that we do have a problem in Europe with clinical trials at the moment. And you'll see some areas where, in fact, the problems exist. The traditional designs that we use to demonstrate efficacy and safety of anti-seizure medication are failing, and I will be discussing that in a moment. Additionally, what's been happening is we don't have validated designs to test anti-epileptogenesis and disease modification. We heard a lot about that today, but when we have to bring that eventually to the clinic, we still lack a validated design. There are shortcomings in clinical trials that have been conducted to date with anti-seizure medication. The traditional design that has brought so many drugs in the market in the last 25 years is what you have summarized in this slide. So basically what we have been doing is establishing a baseline, then randomizing patients in adjunctive therapy to placebo, different dose of the anti-seizure medications, and then eventually tapering or shifting the patients to open label long-term treatment. And these designs served well for many years. They are not serving us so well in more recent times for a number of issues. The first is ethical concerns with continuing to do these trials the way they've been done. There is now a meta-analysis that has clearly shown that patients randomized to placebo in these studies are exposed to an increased risk of sudden death. Second, and that is partly a consequence of the first, it's increasingly difficult for clinicians to enroll patients into those studies. And with so many drugs already in the market, there is virtually no patient that has tried all those drugs. And why should that patient accept to try an untested medication with unknown safety, by the way, and be also randomized potentially to placebo without getting any benefit from the treatment? Then there is an additional issue that comes with that, and that there has been steadily an increasing placebo response in these studies. And this has been happening in many areas of neuroscience and actually more general in medicine. And at least in some cases, it has to do with an increasing number of study sites that are involved into conducting these studies. There is a kind of vicious circle because as it is increasingly difficult to recruit patients, the more and more companies go to remote centers in different countries, including countries that have not so much experience in conducting clinical trials. They increase the number of sites. Not so experienced investigators may not perform at best in conducting the study. That results in a shrinking difference between placebo and active treatment. And the more the difference shrinks, the more we have to go to large number of sites. And this vicious circle eventually kills these designs. Then there have been increasingly concerns about assay sensitivity. And that has to do with the fact that we have found many of these trials design failing to show a difference between placebo and active treatments. And it has to do also with the paradigm that we use in Europe to approve anti-seizure medication as monotherapy, which is the non-inferiority design. And then, lastly but not least, there is a transatlantic divide in regulatory requirements for approval of drugs as monotherapy in particular, and that results in increasing cost and reduces an incentive for companies to work into trials. These are some examples of compounds. They are all in the market, but at least in some studies conducted recently, these drugs could not be differentiated from placebo on a number of studies. And that was not due, by the way, to low statistical power. It has to do to some extent with some of the issues that I've been mentioning before. And then just giving you a glimpse, and I've not changed a single word from what was used in a review of the evidence done by DILE on the quality of randomized controlled trials done in epilepsy over the years. And as you can see, especially those involved new anti-epileptic drugs have been methodologically flawed and cannot answer important clinical questions. And then, more importantly, we do not have well-designed, properly conducted studies for patients with most forms of generalized epilepsy syndromes, in children in general, and of course, in the majority of the rare epilepsy that we heard so much about today. So I close with 2 slides, making some suggestions about how we could tackle these problems. First of all, I strongly feel that we need to improve the way we conduct these studies. And suggestions have been made, for example, new designs that reduce exposure to placebo by using a time to event endpoint. We could and we have to improve patient selection. Getting the correct phenotype and the correct diagnosis is absolutely essential. And this has been remarked many, many times this morning. If we don't get the right phenotype and diagnosis in the trial, the results of the trial will by definition be misleading and the trial will fail. So we need to do absolutely better with that. Then point number 3, we need to, and here is where the community can help, this community in particular, including people working in basic science, we need to design perhaps biomarkers by which we can enrich the population of patients recruited into the trial to make sure that by correctly, again, identifying those that are potential responders, for example, by doing trials by etiology, with treatments that will be targeting etiology, rather than simply seizures, we might enrich the population for responders. And lastly, we need to improve a dialogue among all stakeholders and trying to harmonize the requirements for approval medications across the different regulatory agencies in the world. Just one glimpse on how the time to event design can work. These are examples with perampanel of the analysis post-hoc of perampanel trials. You can see for focal seizures and for focal to bilateral tonic-clonic seizures, you can already find the difference between this is placebo, this is the drug, already within 30 days of treatment. And this is my last slide, which again goes to other solutions. We need to promote, and this is especially to allow more rational prescribing, comparative effectiveness studies that are not designed by the pharmaceutical industry, because they tend to introduce into their designs some elements that will tend to favor naturally the sponsor's product. We need public funding for high quality academic studies. We need to stimulate the creation of networks for doing these studies and take advantage of existing networks, and EpiCare is the obvious example here. We need to exploit opportunities created by electronic health records, but also we need these systems to communicate across centers and across countries. And lastly, we need to streamline the bureaucratic paperwork, and it's not just paperwork, which has become a real incredible hurdle to conduct academic studies. I can see people in this room I've been working with. It has been impossible to conduct trials in Europe because one single regulatory agency in one country refused to accept the study that was accepted by other regulators in other countries. In other examples, it took years of paperwork to get a medicine approved in one EU country used for a clinical trial in another country. So we need to do a lot of work, but we are all here for that. Thank you for your attention.
So, next speaker is David Henschel, who is a professor of molecular physiology and neuroscience at the Royal College of Surgeons in Ireland. He is representing and coordinating EPI Mentor, and I will also read your title, MicroRNA as Targets for Seizure Control and Disease Modification. Please, David.
Thank you very much, Merab. So it's a great pleasure to present some of the therapeutic projects within the EPI Mentor project and give you an update on the progress there. So first of all, just a brief overview of the thinking behind why microRNAs may be therapeutic targets in epilepsy. The first of these was introduced earlier on. It's this concept that these molecules are integrating information within cells, they regulate the gene expression landscape, and a single microRNA can have many targets. So perhaps a multi-targeting molecule is the best way to fix a multi-targeting disease such as temporal lobe epilepsy, where you have multiple causal pathways. The second aspect to argue for targeting microRNAs as therapies is actually to do with the chemistry of these molecules and what happens in the brain and indeed other parts of the body when you interfere with them. So if you introduce an antisense oligonucleotide targeting a microRNA, one injection, it'll knock down the microRNA for as long as a month or 2 months. So this is a very unique and a prolonged mechanism of action, which I think has great promise for a disease such as epilepsy. So there were 4 main sub-projects within the EPI Mentor program which focused on therapeutics. We had a set of microRNAs which we were already interested in, but we didn't yet know whether or not they were important for controlling brain excitability. We then had an unbiased sort of discovery program in which we would screen microRNAs in different animal models, and then we would systematically go through and knock down novel microRNAs to see if we could detect new seizure phenotypes. We then had a microRNA lead at the start of the project, which we wanted to push towards potential preclinical development and overcome some of the translational hurdles, which I'll talk about shortly. And finally, we had a small molecule which an SME within the project had developed rationally that they thought may have anti-seizure effects, and we would pursue that using the consortium. So briefly, 3 of the microRNAs that we began with at the start of the project, we went through targeting with microRNA inhibitors and showed effects either on spontaneous recurrent seizures or on evoked seizures in a canic acid model. We were then also able to identify the probable target involved in explaining that seizure effect. In some of the cases, these were things like ion channels, but we also came across an RNA binding protein, which is a target which is maybe slightly different in the epilepsy field. So I think that was exciting for us. One of the microRNAs at the start was microRNA 135. This has been identified by Jeroen Pasterkamp's group in Utrecht, who had identified it in a screen for microRNAs that regulated axon growth. When we looked at this microRNA within human and an experimental animal model of epilepsy, we saw upregulation of that microRNA just in the epileptic phase. So this isn't being upregulated during epileptogenesis. So in order to determine whether or not there was any functional effect of manipulating this microRNA, we had to wait for animals to become epileptic and then introduce our antisense microRNA inhibitor. So this was something that we had to overcome as an experimental challenge. And these are some of the data. So when we introduce a microRNA inhibitor for this into an already epileptic animal, we see a reduction in spontaneous seizure occurrence and a reduction in spontaneous seizure duration. However, this effect is not permanent. We have, however, a microRNA lead where this effect is permanent. I can't show that data today. Our unbiased discovery pipeline began with 3 different animal models of epilepsy. We extracted microRNA, and then we looked for an intersection between these 3 different models for novel microRNAs. We then picked from that group of 12 microRNAs the ones that hadn't yet been studied in epilepsy, and when we systematically knocked down the microRNA in our in vivo seizure model and identified seizure phenotypes for 3 out of the microRNAs that we targeted and 5 microRNAs where we got a neuroprotective phenotype in the hippocampus. Moving on to microRNA 134, here we already knew that this was a potent regulator of seizures, but we used the network within EPI to test this molecule in multiple animal models of epilepsy. So we were able to show that this microRNA inhibitor reduced seizures in rat models, mouse models, in vivo, and in vitro. And then we turned to some of the translational challenges which this particular therapy approach is going to raise. And the most obvious of all is these are big molecules. How do you get an antisense molecule into the brain? So we studied when blood-brain barrier opening occurred within our animal model of epilepsy and then systemically injected the microRNA inhibitor, timed with when the blood-brain barrier opened so that the molecule could pass through into the target-damaged hippocampus. And when we did this, we virtually eliminated the development of epilepsy in the intraamygdala canic acid model. This is video EEG recordings from 3 months. On the basis of those discoveries and other research within the lab, we were secured a patent. This was granted just in October of last year, and this has now enabled us to leverage industry interest. Here's an example of a collaboration that we began with an American biotech company, Regulus, in which they synthesized microRNA inhibitors, and then we tested them in our in vivo model. And obviously, I can't disclose some of the data, but this was one of the examples of how to exploit that patent. And then finally, we come to our small molecule program. So this was led and driven by one of the SMEs within the EPI Mentor project. They rationally designed a molecule that they thought would have anti-seizure effects. It was then passed over to Stephanie Schorg's lab at UCL, where the electrophysiologist there, Gareth Morris, tested this molecule first in rat brain slices, showing that it reduced seizure epileptiform activity, and then moving to human slices and showing that it also reduced epileptiform activity in resected human tissue. That molecule then passed to our lab in Dublin, where we tested this in vivo in the pentylene tetrazole model and showed that this molecule could delay onset to tonic-clonic seizures in that model. So summarizing, I think what we as a consortium have done is firmly establish this type of molecule as a realistic bona fide potential target for disease modification or seizure control in epilepsy. We have this pipeline of novel microRNAs that we could now move and advance into preclinical development. We've managed to elucidate some of the mechanisms by which these microRNAs are affecting brain excitability, and we're starting to overcome some of the perceived translational challenges with giving this to certain patients. Finally, just to summarize some of the translational and scientific roadblocks ahead, I think we need to take some of these microRNA inhibitors into human cells or into human tissue in order to prove that they have efficacy in a human model, and begin to think about what type of patients or what other forms of epilepsy beyond temporal lobe epilepsy might benefit from this approach to therapy. And finally, as with many of my colleagues here today, some of the key barriers are around funding and cooperation. How are we going to move our molecules and our discoveries into the next phase once these FP7 programs finish? And I think this is what we'll be discussing a lot of today. Thank you very much.
Thank you, David. So I will introduce the next talk. It is given by Sergius Joswiak, and he's a professor and head of the Department of Pediatric Neurology at the Warsaw Medical University. He's representing and coordinating EPISTOP, and the title of the presentation is Pre-seizure versus post-seizure treatment of epilepsy, pros and cons. Please.
Thank you very much. And my second trial with the pilots, I hope I will manage. Yeah. So, tuberous sclerosis is a model of epilepsy and epileptic encephalopathy. I think it's a quite good model. It's a genetic condition with autosomal dominant mode of inheritance, with very high incidence of epilepsy, so up to 90% of patients develop epilepsy during lifespan. There is a very high incidence of post-epileptic intellectual disability and autistic behaviors. In general, in population of patients with tuberous sclerosis, it's about 50%. There is a precise window of epilepsy onset. It's very important. In something like 70% of patients below 2 years of age develop epilepsy. There is a relatively known mechanism of epileptogenesis, mTOR disinhibition. And now, during the recent years, we have a possibility to diagnose prenatally. We learned how to do that. How to diagnose tuberous sclerosis prenatally. So we have access to the patients. So it's for the first time, as a neurologist, we have patients who will develop epilepsy before epilepsy. So we may look at those patients before epilepsy. So it's a very unique opportunity. Of course, epilepsy doesn't start at the moment of clinical seizures. So now we know from different studies, a lot of animal studies, that before the appearance of clinically seen obvious seizures, we have a lot of different events at different levels of tissue and molecules, so leading to these clinical seizures. So in 2011, also in the very nice review on infantile spasm treatment review written by Karl Staffström at this moment, and in 2011 he was also the main editor of Epilepsia. It was in this paper, a review paper, he also considered this preclinical treatment of epilepsy there is an onset of infantile spasms, and there is a usual time of treatment initiation, and there are some arrows just indicating possible interventions to reduce epileptogenesis. So at this moment, we started to talk not about anti-epileptic, but about anti-epileptogenic treatment. Of course, with all these issues related to the drug evaluation, which are anti-epileptic or which are anti-epileptogenic. Oops, it's not this pilot. Okay. Okay. So, of course, we need to come back again to the animal studies, and we learned that the treatment before clinical seizures is available and may be successful in animal models. One of such studies were studies done in tuberous sclerosis in animals with TSC1 knockout mice. These animals are demonstrating epilepsy, encephalopathy, premature death. And in this study, these animals were treated with rapamycin, so mTOR inhibitor, which was given at the age 14, at the day 14 after delivery with rapamycin as early treatment. And these animals didn't develop epilepsy nor premature death, while those who were treated as a late treatment with rapamycin at 6 weeks of age still developed epilepsy. There was possible suppression of seizures and also prolonged survival when compared with the with animals with the knockout. So from this study, we also learned that it is possible to introduce it to clinical practice. I mean, in animals, in animal clinical practice, this prevention of epilepsy or amelioration of this epilepsy. So, how to translate this knowledge into patients? In tuberous sclerosis, we know that the main substrate for epilepsy are cortical tubers. There are multiple cortical tubers, and some of them are epileptogenic, producing abnormal EEG. If these epileptogenic tubers are still producing abnormal EEG, some patients will develop seizures. If they are not treated, drug-resistant or appropriately or effectively treated, they develop drug-resistant epilepsy. And all these aspects are producing intellectual disability in patients with tuberous sclerosis, which we may observe, particularly in these very, very young patients. In 2011, Paolo Curatolo also wrote a very interesting paper just demonstrating that in 10 children who were treated in the first week after the onset of seizures, so very early, and were observed up to 88 months, he documented with this very early treatment that 50% of patients were seizure-free. There were no severe forms of intellectual disability. Still, 5 of them who had moderate intellectual disability. So we were thinking about even earlier treatment of epilepsy, not when we have seizures, but when we may identify patients with very high risk of development of epilepsy and to treat at this stage. In 2011, we published our report on the comparison of 2 groups. Standard treatment group, treated when they had seizures, and preventative treatment group. This was not a randomized trial, it was a historical group when we were able to diagnose these patients very early, prenatally, or just in the first months of life. So in both groups, we had patients diagnosed with TSC below 3 months of age. But these patients were followed up to 24 months, and this group also up to 24 months. This without treatment. They developed seizures, and we started to treat them. And we, in this preventative group, we did EEG every 4 to 6 weeks. And when there was abnormal EEG, when we have polyspikes, not the single spikes, but polyspikes activity, synchronous activity on EEG, we started to treat them with the normal treatment. Why? So we are not talking about the new drug. We are talking about the drug which is usually used for infantile spasm in tuberous sclerosis, and we know it's very effective, but we use it earlier than usually. And it is interesting that in both groups of patients, we had exactly the same number of those who were treated. In the standard group, they were treated because of the clinically obvious seizures. In this group, they were treated because of abnormal paroxysmal activity on EEG. And we had very interesting results because in the standard care group, at the age of 24 months, there were only 2 patients with normal EEG at 24 months, while in this preventative group, we had a normalization of EEG in the majority of those patients. Only 2 patients had abnormal EEG at the age of 24 months. And again, comparing the number of patients with drug-resistant epilepsy, we had a lower number of those patients. We had much more patients with seizure freedom at 24 months. We had better IQ score, and what was at that moment the most important for us, we had no patients with severe forms of intellectual disability. So this study was the beginning of the EPISTOP project, and there were several presentations now, so I'm not talking about that. The results of our study was also confirmed 2 years ago by American group. They also analyzed the EEG as in these young infants, in newborns, who they did similar study also. They did sequentially EEG and observed the appearance of seizures in those who developed abnormal EEG. Because of this study confirmed our publications from 2011. So soon, several months later, in December 2016, they launched in the United States PREVENT trial. Also, it's a trial when the American researchers are going to treat with vigabatrin those patients before clinical seizures. And this is my last slide. What is the possibility with this procedural treatment? I think that we have a possibility to reduce number of patients with epilepsy and epileptic comorbidities, particularly drug resistance, intellectual disability, and autistic behaviors. Paolo Curatolo will present also results of our study EPISTOP when we are talking about this manifestations of epilepsy in TSC patients. And it would be interesting to identify another groups of patients. I think this is a future if we are able to identify these patients with very high risk of epilepsy development. Particularly, we are talking about a very sensitive brain. I mean, these infants below 2 years of age. And if there is such poor outcome in these patients, maybe we should consider this preventative treatment in this particular group of patients. What it's cons, it's a safety. Safety, it's a clue, it's a very important issue in these patients, very young patients. So this is something we need also consider just planning future studies in our patients. Thank you very much.
It's a pleasure for me to now invite up to the stage Wolfgang Löscher, who is the professor and director of the Department of Pharmacology, Toxicology, and Pharmacy, as well as the head of the Center of Systems Neuroscience at the Veterinary University in Hanover. Many years ago, he invited me and one of our young researchers to his lab and helped us establish experimental epilepsy surgery, research on our own. So thank you for that. Thanks for the talk today on multi-targeted drug combinations.
Thank you, Felix. So, as you are all aware, there's no preventive treatment for patients at risk to develop epilepsy that is approved for this treatment in patients. And this is despite the fact that more than 30 years of research has been performed on epilepsy prevention. So we thought that one explanation for this might be the complexity of epileptogenesis. And some 5 years ago, we proposed, therefore, that targeting multiple targets in an epileptogenic network, so-called network pharmacology, may be more effective than treatment with single, highly specific compounds as done in the past. So in order to facilitate clinical translation of such a network approach, we used clinically approved drugs that we repurposed for trials on epilepsy prevention in animal models. And this approach, so a combination of drugs with different targets for anti-epileptogenesis, is performed by my group in one of the work packages of the EPI Target Consortium. So as I mentioned, we proposed this approach 5 years ago in a Nature Review article, and when we did that, we were aware that this approach was used in other indications over a couple of years, as for instance reviewed in this Nature Chemical Biology review, where they had different examples, neurological diseases, other diseases, and there was one clinical entity, a rare neurological disease, where this network pharmacological approach worked in patients. So this prompted us to try to, in a way, translate this network approach to treatment or prevention of epileptogenesis. And what we did was a relatively simple approach. We decided for a number of potentially critical alterations during epileptogenesis, developing after an epileptogenic brain insult, including neuroinflammation, neurodegeneration, neural hyperexcitability, and others. So in order to interfere with neuroinflammation during epileptogenesis, we chose a number of anti-inflammatory drugs, in addition to that also a number of antioxidant drugs. In order to interfere with the development of neurodegeneration, we chose drugs that affect either regeneration or plasticity, or both. In order to interfere with the development of neural hyperexcitability, which of course is a hallmark of epileptogenesis, we chose different GABA-potentiating and or glutamate suppressing drugs. And finally, we chose a number of drugs with presynaptic effects. Now, altogether, we decided to use 17 drugs in 12 different combinations. And I decided, being a pharmacologist, to perform the preclinical trials in animal models in a very similar way to clinical drug development. So we subdivided the development into different phases. Phase 1 being pharmacokinetics and tolerability in healthy mice. And the advantage of using clinically approved drugs is that the pharmacokinetics are known in rodents and can be used from the literature or other data files. However, the tolerability of the combinations was, of course, not known. And so this had to be determined in phase 1 to de-risk the efficacy trials. So the table there is just an example of the pharmacokinetic differences for a number of the compounds that we use between rats, mice, and humans, just to remind you that most drugs are much more rapidly eliminated by rodents than by humans, thus necessitating the development of relatively complicated drug treatment schedules with several times daily application or even continuous drug application in rodents. So the next step then was to translate the clinical trial design into preclinical trial design. This is a two-stage approach which was mentioned in the title of my talk. So the first stage is mice. And in phase 1, which I mentioned, we use relatively small groups of animals for testing the tolerability of the compounds in healthy animals. And if a combination is tolerable, Then we go into animals in the latent period after a status epilepticus induced by intrahippocampal kainate, because it is known that epileptogenesis may alter the tolerability of drugs. So if the combination is tolerable in these animals, then we go to proof of concept trials in the intrahippocampal kainate model, which I will show in a moment, to test whether the combination is anti-epileptogenic, and for that we have to increase the power of the study, so we use relatively large group sizes for this part. If the combination in mice turns out to be effective, then we go to the second stage, which is rats, to have a second species then, and we try then to reconfirm the anti-epileptogenic efficacy of the drug combination in rats in a status epilepticus model, and if this turns out to be positive, then we plan to go into another model of acquired epilepsy, for instance, a TBI model. So this is the current situation with all these combinations. All of them has been tested for tolerability, and at the doses which we combined, all the combinations were tolerable both in healthy animals and during the latent period. And then we started with the efficacy trials in mice, and one of the combinations that we tested, combination of levetiracetam and topiramate, proved to be effective, which I will show in a moment, whereas two other combinations did not have any anti-epileptogenic effects, and there are posters on both examples, a negative example and a positive example. The reason for having levetiracetam in most of these drug combinations was merely the fact that this is one of the few novel anti-epileptic drugs for which there have been clinical epilepsy prevention trials conducted by Pavel Klein in Bethesda, Maryland, and he is our cooperation partner. So just to illustrate how such a drug trial looks like, we induce epileptogenesis by intrahippocampal injection of kainate. The latent period in the mice which we use is about 5 to 7 days. So this is a therapeutic window of opportunity, followed by development of epilepsy with spontaneously recurrent electrographic and electroclinical seizures. For the levetiracetam topiramate combination, we conducted 7 separate trials. One with levetiracetam plus topiramate, of course, which we repeated twice, and then treatments with either drug alone. And this was started 6 hours after the kainate injection and went over the whole latent period. Then at different times thereafter, we performed continuous EEG video monitoring to determine the spontaneous seizures. In addition to that, we used multimodal brain imaging with both microMRI and microPET to determine drug effects on blood-brain barrier integrity, on neurodegeneration, and on inflammation. And then finally, the animals were killed at different stages of the process for both brain histology and immunohistology and gene expression analyses. So this was the first experiment with a combination showing a relatively pronounced effect of the combination versus vehicle. In each trial, we had concurrent vehicle controls, and we repeated the experiment and confirmed the experiment and put the data together in a highly powered study showing that there is a highly significant effect and that a number of the animals do not develop epilepsy at all because of the drug treatment. Either drug alone did not have any effects. Then we looked for potential mechanisms of the combination. We found no neuroprotective effect. MicroPET and microMRI imaging did not indicate any anti-inflammatory, blood-brain barrier protective or neuroprotective effect of the combination, so that we decided to cooperate with Michael Johnson, and he's currently performing gene regulatory network analysis to identify the potential therapeutic targets of the combination. Challenges, bottlenecks. So of course, testing high numbers of drug combinations for anti-epileptogenesis is extremely laborious and time-consuming and expensive. And one additional problem here is the lack of reliable algorithms for automated e.g. seizure detections in the animal groups. As a consequence, we cannot test different doses of the drug combinations, but have to rely on one specific dose combination, which of course may lead to false negative findings. Next steps are, of course, that we continue with the testing. And the conclusions are that the algorithm seems to identify potentially, the testing algorithm, potentially interesting drug combinations. And we will try to translate these combinations to other animal models. And finally, I wish to thank my colleagues and, of course, EpiTarget for the help in performing our studies. Thank you.
Thank you very much, Wolfgang. So we have our last but not least talk by Marco Ledri from Lund University, Sweden. And he will be representing epiXchange, not this one, but the previous one.
Hi everyone, thank you Merab for the introduction. So what I will talk today about was a project that we did within the epiXchange consortium, and it deals with the effects of neuropeptides on excitatory transmission in human epileptic hippocampus. So just a very brief introduction on neuropeptides, and especially on two of them, NPY and galanin. Those are two neuropeptides that are typically co-released with neurotransmitters in the brain, and they've been shown several times to have seizure suppressant properties. And the mechanism of action of which these peptides are supposed to work is to reduce presynaptic glutamate release. And they've been tested in several animal models of epilepsy, of epileptogenesis. They've been tested also in vitro, in slices. And what one typically does, especially in the animal model, is one delivers these peptides via AAV viruses, express them in the epileptic focus, and then tests the EEG and so on, and see what are the effects on the seizures. And because of all these positive effects of these peptides, they have been proposed as potential candidates for future clinical trials in humans. But what Merab also used to say was that usually the best model for a cat is a cat, and preferably the same cat. So if one wants to see the effect of these things in human brain, we have to look at what they do in the actual tissue where they're supposed to be targeted later on. And that was something that wasn't known. So what are the effects of these peptides on human pharmacoresistant epileptic tissue, which is the actual target? And to do that, we gathered tissue from 13 patients that undergo selective surgery to remove the hippocampus, which was the focus of the seizures. All the patients were diagnosed with pharmacoresistant temporal lobe epilepsy, and all of them, they had hippocampus sclerosis. And here in this figure is just, I don't know which one it is, this one. It's just to show an example of how the tissue looks like. There is sclerosis in the dentate gyrus, there's loss of neurons in CA1, and there is sprouting of excitatory fibers throughout. So what we do with the tissue, we get it from the surgery room, and then within 1 hour, we bring it to the lab. We slice it with a vibrotome, keep it very cold in the beginning, and then slowly warm it up. And we transfer it to an electrophysiology setup where we can do both single cell recordings, whole cell recordings, or we can do field recordings to monitor the activity of groups of neurons. And we applied MPY and galanin separately in the bath during the electrophysiological recordings. This is an example of a cell patched from the tissue. I think this was a dentate granule cell. I'm sorry. And as a first step to verify that the tissue is viable before we proceed with the experiment, we usually just patch a cell and induce action potentials or monitor synaptic transmission, or like the appearance of spontaneous EPSCs or iPSCs, just to see that the tissue is viable and we can proceed. And next, we move to the actual experiments where we applied MPY and galanin to the tissue. This is an example of a field recording, which is done in CA1. So what we do, we stimulate presynaptic fibers, and then I think in the stratum radiatum, and we record from the same stratum radiatum. This is how the responses look like. This is the stimulation artifact. This is a presynaptic fiber volley. This is the actual field EPSP. And we did a protocol where we had an initial stimulation, and then after 50 milliseconds, we had a train of 10 stimulations at 40 hertz. We record a baseline period of about 15 minutes, and then we apply the peptides, 1 micromolar or 0.5 micromolar to the bath, and then we monitor what happens to the field EPSPs over time. And when we apply galanin, basically nothing happens. The responses stay very constant and very stable throughout the whole recording. But when you apply MPY, you see a pretty dramatic 60% decrease in the slope of the first response, and a decrease in most of the other responses of the train as well. And importantly also, we see a difference in the per pulse ratio, which is the ratio of the slope of the second pulse versus the first pulse, which increases, which indicates that the drug and the MPY is acting presynaptically and altering glutamate release. As a last step to confirm that these are glutamatergic responses, we apply MBQX, which is a block of AMPA receptors, and we can eliminate completely the response to make sure that this is what we actually wanted to record from. So then we did a new set of experiments very similarly, but we changed area just to confirm that the effect is not specific for CA1, but it's a more general effect that happens throughout the human hippocampus. So we recorded from the dentate gyrus, we stimulated the molecular layer, and recorded from the molecular layer, and the experiment is very similar. Here also galanin didn't do anything, failed to alter whatever slope of, so there was actually a very minor effect, but MPY has a much stronger effect, and again the per pulse ratio was increased, indicating that the mechanism was similar as in CA1. So what these results show was basically that what we saw in the animal models doesn't necessarily reflect what we see in the human hippocampus. And there were a number of challenges, and there still are a number of challenges for this type of approach. First of all, at least in our center, the availability of human tissue is rather limited. I know in some other centers there's a bigger availability, but since this tissue is very valuable for us, for this type of investigations, we were wondering if we can extend the viability of this tissue over time to do an increased experimental output, to record from more slices during the same. Now we were just basically doing it over 18 hours after the resection. And the second thing that was missing for us was the possibility to induce epileptic formativity. Here we just monitor glutamate release, but we don't know if the peptides are actually affecting epileptic formativity in these slices. And lastly, we want also to develop a method to test viral vector approaches in this type of tissue, so which will bring us even closer to the clinic sort of translation. To do all these things, and especially to increase the viability and the output, we developed lately a different way of slicing, maintaining, and incubating the tissue, so we can, instead of only having it available for 18 hours, we can now record it up to 2 or 3 days after the resection with this with this new method. And at the same time, we changed the recording parameters where we have a dual superfusion recording chamber instead of the slice being perfused only on the top side, now they are perfused in the rig on both sides. That increases oxygen availability, and that increases neuronal activity and network activity, so we can reliably now generate epileptiform activity on this type of tissue. And as the last, this is the last slide, I would say. We want to propose this type of platform and use this platform and propose it as a sort of part of a roadmap towards clinical trials for gene therapy in epilepsy. So we want to emphasize that it's since there was this difference with galanin, what we saw with galanin in the human tissue versus the animal models, we want to emphasize that it's really important to test potential strategies or therapeutic strategies on this type of tissue, especially the ones that are modifying synaptic transmission, or that are supposed to modify synaptic transmission. So to have a confirmation, and if they don't work, for example, in the example of galanin, we would probably stop the clinical trial, so stop the progression of that particular strategy towards clinical trial. While in the case of MPY, where we see a very clear and strong effect, then that will be an indication to continue. And thank you very much.
Thank you, Marco. Do we have time for questions? Yeah, 10 minutes. So please, speakers, if you would like to join us here. So the session is open for questions and comments, please.
Thanks, Merab. Not surprisingly, my question is to Emilio. Where is he? There he is. I've always thought somehow or other that N=1 trials have been neglected, particularly for rare diseases. When you can have a placebo and a treatment effect, you can randomize the order, you can do it 2 or 3 times, and you can get data on a number of patients individually, which you can then add up to hopefully something worthwhile that the regulators will recognize, although I suspect they won't recognize it yet. Any thoughts?
Do we have more microphones?
Well, I agree, Martin. The N=1 trial is one of the ways by which we can do trials in certain situations. And there are a number of tools which have been perfected to conduct studies in small number of patients. So definitely, that would be one of the ways to go. And we had examples, by the way, of very successful trials that were even accepted by regulators, done with 20 patients overall. And then if you look at the results of those studies, they could have found a difference in even a smaller number of patients. It's just a matter of, as mentioned, of enriching the population. So if you really have a very good target population for a good compound, you don't need a large population to get the magnitude, to demonstrate the effect size that is there. Agree.
Thank you. More questions? Anna Maria?
It's just a comment about disease modification treatments in the clinical setting. So we use these treatments in the animal models when there is epilepsy diagnosis in the animals, when they develop the first spontaneous seizures, and we can modify the disease. However, in the clinical trials, I understand that most of the cases, the novel drugs are used in chronically drug-resistant patients that are in a very end stage, so to say, of the disease. So I wonder whether to test novel disease modification treatments, one should think about a different clinical approach.
Again, I fully agree, and I guess at least for disease prevention, the solution would be through biomarkers. Because until we just look at a large population of patients and the overall probability of those developing epilepsy is between 10 to 20%, you'll get nowhere. But we can identify those at the beginning, and what you have shown this morning indicated there may be a way to do that.
But also after epilepsy diagnosis, maybe
after
to
epilepsy
test
diagnosis,
novel treatments at that stage, not when the patients...
then you may have go for different paradigms. For example, not looking necessarily at seizures, but also other indicators of disease modification, such effect on comorbidities, intellectual development as well. But you can also use models by which you look long-term at seizures. The everolimus trial gave us a hint of how we might be looking at that on the long term, even for seizures, because as you remember the effect, at least in the initial trial, when the drug was a steady state, there was a steady decrease in seizure number over time. So there may be ways by which that can be shown. What I just said is that at the moment, no one has actually demonstrated in the clinic disease modification for certain, but that's the way to go.
More questions there?
I would like to ask Professor Lesher regarding the network pharmacology, which I quite like the idea. But is there this idea also that using a combination of drugs one should also use like lower doses of each drug? Or could you make a comment about this, please?
The mic. That was the reason of my comment on different doses being tested. The problem is we need at least 6 months for one drug combination, at least. And then we have to replicate it to increase the power of the experiment, so it's another 6 months. If you would decide, which we currently think about, if once we have an effective combination, then we could think about changing the dosages, because you are right. The network idea is that you can use much lower doses than those which are used in monotherapy. But that for me would be the next step, because you do not know that in advance whether by decreasing the doses you will get any effect. And if you have a negative data set, then to convince the co-workers to do another laborious experiment with higher doses is not easy. So that is our very pragmatic approach.
So, no more question here? Microphone, please.
Maybe while I'm giving the microphone, I can ask my question. Also, for Professor Löscher, you are, as you say, it's very labor-intensive to test all the treatments. But wouldn't that be a kind of an initiative for many labs to study this? Not only your lab doing this, but sure, it could be a collaborative effort. Many people work with the same model. Many people have the capability of recording from mice. So shouldn't this expand beyond your laboratory and then increase?
Right. At least in academic labs, you very often have your favorites. So one lab is studying mainly inflammation, another lab is studying whatever, microRNAs. So it's not easy to convince other groups to use a similar approach. But one example is here in the room, which is Anna Maria Vezzani's group. She's now also testing combinations of drugs, but with a slightly different approach, which you just mentioned. She's starting treatment after onset of epilepsy, whereas we are starting before onset of epilepsy. But you are absolutely right. That would be a necessary step to combine efforts.
And as a follow-up to this question about feasibility, would implementing AI and some system analytics there help in the process by narrowing down the number of throughput screenings that you have to do in the combinations?
Yes. And what I did not mention, EpiTarget also looks on biomarkers, so we also look on biomarkers in our experiments. And hopefully we can enrich by biomarkers, but at the moment it's not possible to do this. But yes.
So to get Wolfgang off the hook, I have a question to Fabio. Fascinating data and technological development. So I was wondering in terms of NPHR, halorhodopsin, whether long-term activation would be any concern in light of the recent publications that came out, that also from our lab, showing that it may generate some aberrant activity in the neurons.
Well, we are still at the stages in which it's difficult to evaluate if there will be such an effect. But for sure, the inhibition is, the inhibition will go on only upon acidification. So it will not be a strong, prolonged effect as you can achieve with continuous illumination as we do in the lab when we use halorhodopsin. So this is the idea. Well, the pH changes by Q-culling, we checked in vitro. They last until, of course, the hyperexcitation goes on, and then the pH slowly goes back to neutrality in, let's say, tenths of minutes, like 5-10 minutes. Yes, yeah. No, that's a concern. Yeah.
Okay, more questions?
excuse me.
Yeah.
The most prevalent human occurring model of epileptogenesis would be post-stroke or TBI, post-TBI epilepsy. Where are we here in this field to look how epilepsy develops in humans, not say only in animal models, to look like TSC, which is kind of an orphan disease or a rare disease. Stroke is so prevalent, TBI is quite prevalent, so where are we here in this field?
I think that epilepsy is rather a symptom, it's not disease.
Okay.
So in fact, that's why we have chosen tuberous sclerosis as a very homogeneous group of patients. And I think still the results from TSC studies, it will be not easy to translate to the adult patients, because the adult brain, it may be a completely different mechanism of action. So now we are going to continue the similar studies in another group of patients, but still in the same age group of patients, because it's very sensitive, it's very peculiar brain. So I think, and besides, we have a lot of epigenetic factors in adult patients with post-stroke. So we wanted to avoid that.
Okay, more questions, comments? A microphone is coming, yeah.
Sorry, good afternoon, everyone. My name is Anna Grassa. I'm a program officer at the European Commission, DG Research and Innovation. I don't have a question, I have a comment. And I would like to thank you, all of you, for raising some of the important issues, especially while conducting a clinical trial. In 2016, the European Commission established an expert group called STAMP, which stands for Safe and Timely Access to Medicine for Patients. And actually, some of the issues that you raised today are in focus of this group, which I'm also a member of. So hopefully, together with the member states and representatives of pharmaceutical industry, we can hopefully overcome some of these challenges. Also, the Commission in April 2018, this year, closed a call for proposals for coordinated support action to strengthen the regulatory advice, access to regulatory advice for researchers in academia. So some of these issues will be also raised tomorrow at the workshop, but we are working on these challenges, and hopefully together we will overcome them.
Okay, thank you. I'm afraid we have to stop this interesting discussion because of the lack of the time, and I would like to thank all the speakers and all of you, and also give Felix, you want to say something? No? Okay, so thank you. Thank you very much.